Hello and welcome to all my dear students. I hope you are doing fine and must be studying well at home. So finally we are going to start with the new chapter of class 12th, very important tricky chapter, that is Principles of Inheritance and Variations. This chapter is also called as Genetics Part 1. In this chapter, we are going to study the pattern of inheritance of genes, that how genes are inherited from the parent to the offspring. What are the laws that genes follow while inheriting? So yes, this chapter is very interesting, tricky also, and sometimes students get scared of this chapter also. So now, no more such scare kind of feeling with respect to this chapter. We are going to make this chapter very easy and simple. Now, today beta, since it's the first class, so it's a kind of introductory class where I will be teaching you first some technical terms, some terminologies which I will be frequently using in this chapter from the beginning till the last. So that's why I chose that why not to explain all the technical terms beforehand and then we will start the chapter. Okay? Yes or no? Chalo. So let's start with today's class on Principles of Inheritance and Variations. The chapter is also called as Genetics Part 1. So what is genetics? How will you define genetics, beta? Genetics is basically the branch of science which deals with the principles of inheritance and variations. So it is the branch of science which deals with heredity and variation, that how the genes are being transmitted from the parents to the offspring. If anybody is studying in this branch, then this sub-branch of science is called as genetics. So genetics is the branch of is the branch of science which deals with the principle of variations and inheritance, which is actually called as heredity. So what is heredity? Heredity simply means transmission of characters, transmission of genes from the parents to the offspring through gametes, right? You are produced because of the gametes. Father donated sperm, mother donated egg. Those egg and sperm are actually carrying the chromosome from the parents. Those chromosomes are having genes, and genes are the heredity units. These genes basically are what inherited from the parents, and that because of the same genes we look similar to our parents. Correct? So how will you define heredity? Heredity is the inheritance of characters. Characters means genes. You know, genes regulate characters. So someone says that, okay, my eyes is similar to my father, my nose is similar to my mother. Why? Why my eyes and nose are similar to my parents? Because these characters which I have got, maybe my brown eyes, maybe my pointed nose, all these features, all these characters I have got from my parents in the form of genes. So genes are basically regulating the characters, and the transmission of such character is known as heredity. The branch of science which deals in with heredity is known as genetics. I hope all the two terminologies are clear. Now, who is known as the father of genetics? The father of classical genetics is Gregor Johann Mendel. He was the first person who actually could explain few laws of inheritance. So he's also known as father of genetics. Who's known as father of genetics? Gregor Johann Mendel. Now, before I enter into the history of Mendel, as I already told you, first we are going to discuss some terminologies. The first and very important terminology which I am going to use again and again is genes. Now, everybody says, okay, genes, these are the genes which I have inherited from my parent, that's why I look like this. But how will you define basically gene? What do you understand by the term gene? Beta, genes are those active part of the DNA, genes are those active part of the chromosome which are transferred from the parent to the offspring. These are responsible for controlling the expression of every character. Suppose I have brown eyes, I have black hair, I am tall or short, whatever my body is showing characters, it is all because of the genes. So genes are the regulatory aspect of the chromosome which controls the character. Chemically, genes, they are made up of DNA, right? Genes can also be called as unit of heredity. So how will you define genes? Genes are basically unit of heredity. What are genes? Genes can be defined as beta unit of heredity transferred from parent to offspring. Second thing, these are chemically made up of DNA. So sometimes in some books you will find that DNA is the unit of heredity, but actually DNA, genes, they are almost one and the same terms. So basically genes are the unit of heredity which are made up of DNA. Genes, they express themselves, they produce protein in order to regulate the character in an organism. So third point you can write for genes. Genes, they expresses themselves. Genes expresses themselves to regulate a specific character in an individual. Correct? So these genes are responsible for regulating the character. Now, where are the genes located? So genes are beta located on chromosomes. Genes are located on chromosomes. Chromosomes we know. Okay, in a diploid cell of a human, 46 chromosomes are present, 23 pair. That you know, in 11th you have already studied the cell chapter and cell cycle chapter. You know what is chromosome. So suppose this is what chromosome actually is. Chromosome is basically condensed form of chromatin fiber, right? Now this condensed form, suppose it is a diploid cell. So diploid cell will have pair of chromosome, right? Now these are suppose homologous pair. These are homologous pair in a diploid cell. Homologous means chromosome which are exactly same in shape, size and morphology. Now beta, every gene has its specific location on specific chromosome. Like for example, suppose my gene 1 is responsible for controlling eye color and it is located on chromosome 1. So for every Homo sapien, there will be gene 1 controlling eye color located on chromosome 1. However, if you change the species, if you bring some other species organism, then in them, the gene 1 nature will change, its position in the chromosome will change. However, within a species, the function of gene, the location of gene on chromosome is fixed. Right? So what will you write the next point? Genes are located on chromosome and those locations are called as loci. These locations of genes on chromosome are known as loci. Correct? For each gene, there is one loci located on a pair of homologous chromosome. Suppose this is a homologous chromosome. Correct? Suppose this is chromosome 1. This is set of chromosome 1. Chromosome 1 is having gene 1. Correct? So this overall is known as loci 1 having gene 1. Correct? On a homologous pair, for one gene, there is one loci like this. This is not loci. You can't say that this is okay. This is loci 1 and this is loci 2. No. On a homologous pair, there is only one loci and one loci is representing the position of gene 1. Loci 2, gene 2. Loci 3, gene 3. Correct? Yes or no? Understood everybody? Okay, so what did I said? Genes are located on chromosome. These locations are called as loci. Loci is a plural term. Locus is a singular term. For each gene, there is one locus on a chromosome. Chalo. Now beta, in a diploid individual, each gene is represented by a pair of alleles. So next terminology after gene which you have to understand is alleles. Alleles are called as alternating form of a gene. Now, if I talk about a diploid individual, in a diploid individual, each chromosome is represented by a pair. Like, right? Yes or no? So, this is my one chromosome pair. Correct? In a diploid cell, this is my one chromosome pair. In this one chromosome pair, for each gene there will be one loci. Suppose this is loci or locus, not loci. This is locus 1 having gene 1. Now, in a diploid organism, this gene maybe which is called as T gene will be represented by a pair of allele. Maybe capital T, maybe small t. Correct? So this capital T and small t, these two are beta alternating form of T gene, right? Which are located on the same loci on a homologous chromosome. It's not like that. Suppose this is one homologous pair, this is another homologous pair. So if one of the allele of the T gene is here, then another allele is here. No, not like this. Alternating form of a gene is located on the same homologous chromosome on the same loci. Correct? Now, so what are gene alleles? Alleles are basically beta, they are the alternating form of genes. In a diploid individual, one gene is represented by two alleles. However, in a population, like for example, beta, if I talk about 5 people, if I talk about 5 Homo sapiens, so 5 Homo sapiens together, they are making a population, correct? So in a population, one gene can be represented by more than 2 alleles. However, in an individual, there will be only 2 alleles. This is known as multiple allelism. Now, what happens in blood group? Blood group is controlled by I gene, correct? Which is having 3 different alleles in a population. The 3 different alleles of I gene are IA, IB and recessive I. Correct? Now beta, these are 3 different alleles. 3 different alleles of a gene I, which is responsible for controlling blood group in humans. Now this is for population. Now suppose if I talk about myself, I am one of the Homo sapien, I am also having I gene. But in myself, in my body, because I am a diploid organism, my body is represented by only pair of homologous chromosome. So for I gene, there will be only 2 alleles out of the 3. All the 3 alleles cannot be present in a diploid cell. Out of 3, only 2 will be present. Correct? Like for example, I am having IA, IB. Both the alleles can be same also. Both the alleles can be different also. Correct? So this is the loci. This is locus 1, gene 1, that is I. And that is represented by 2 alternating forms IA, IB. So IA, IB are the alleles of gene I located on the same locus on a homologous chromosome for a diploid cell. However, if I talk about population, now suppose I am one Homo sapien, my combination is IA, IB. You are another Homo sapien, then your combination may be IB, IB. There may be third Homo sapien whose combination may be IA, IO. So different individuals, we together we make the population. In a population, a gene can be represented by more than 2 alleles. However, in a diploid individual, that gene will only be represented by 2 alleles, any 2 allele. They can be same, they can be different. This concept is known as multiple allelism. So what is allele? Allele is simply the alternating form of a gene. Correct? Next, multiple allelism. More than 2 alleles of a gene in a population is called as multiple allelism. Example, blood group. However, a diploid individual will only have 2 alleles for a gene. Will only have 2 alleles for a gene. Now suppose if an organism is haploid like gametes, so there only 1 allele for a gene. Diploid, 2 allele for a gene. Triploid, 3 allele of a gene. So depending upon the number of chromosome, the number of alleles of a gene will be present. Understood? Okay. Now beta, alleles can be of 2 types. Either dominant allele or recessive allele. Now what is dominant? What is recessive? Dominant means the allele which is the allele of a gene which is capable of expressing itself under all conditions. The allele which is capable, which is responsible for producing proteins and deciding the phenotype of an organism. It is actually the dominant allele of the gene which is controlling the character of an organism. Correct? On the other hand is the recessive allele. Allele which do not express, allele which is not responsible for producing proteins. Like for example, beta, T gene. Now, this T gene is responsible for controlling the character height. Right? Now, there are 2 alleles possible. One capital T, one small t. Now, suppose an organism is having a diploid organism is having the 2 alleles which are same, capital T, and there is another diploid organism which is having both the alleles as recessive. Now what will happen? Here, both are the dominant alleles. They are going to express themselves. They are going to produce proteins which are going to produce the production of, which is going to increase the production of growth hormone, and hence the body is going to become tall. So organism having this combination will appear to be tall. However, if the combination is of second condition, if both the alleles of a gene in a diploid organism are recessive, in that case, no protein is synthesized, neither recessive alleles, they cannot produce protein. So neither this is going to produce nor this is going to produce any protein. If no protein, then no enzyme. No enzyme means growth hormone is not able to get produced in large amount. Hence, the organism will become dwarf. Right? So, beta, you cannot say, remember one thing, small t, recessive allele is not responsible for dwarfism. Actually, absence of dominant allele is responsible for dwarfism. What I want to convey is there are 2 types of alleles, dominant and recessive. Dominant alleles are those alleles which are able to express themselves under all conditions. They are responsible for controlling the character in an individual. Absence of dominant allele means presence of recessive allele. So, if dominant allele is not there, that means a specific protein will not be produced. If specific protein is not produced, then definitely the phenotype of an organism is going to change. So, one example I took over here is for T gene. Now, in T gene, beta, there are 2 types of allele, capital T, small t. Capital T simply means the allele which is dominant, which is capable of regulating the growth hormone. It can increase the production of growth hormone, so the individual will appear tall. However, if an individual is not having any capital T, having only recessive T, that means no protein synthesized, no growth hormone synthesized, hence organism will become dwarf. So dwarfism is not the character, is not the trait coming because of small t. It is due to the absence of dominant T. If dominant T present, tall. If dominant T completely absent, dwarf. So it is actually the function of dominant allele to regulate the phenotype. Recessive allele is just the one which is completely inactive, no protein synthesis. Understood? So how many alleles can be there? Two types, dominant and recessive. Dominant alleles which express itself under all conditions. Recessive allele which do not express itself under any condition. Now, after this beta, okay, in a diploid individual, you know, an allele, a gene is represented by a pair of allele. Now, both the alleles, if they are same, like over here, capital T, then this combination of allele for a gene is known as homozygous. And if both the alleles are different, then it is known as heterozygous. What is homozygous? When both the alleles are same for a gene in a diploid organism, like capital T, small t. Opposite to is heterozygous. Heterozygous means what, beta? When both the alleles of a gene in a diploid organism is different, like this. So this is your heterozygous condition. Got it? Yes or no? Now there is one more condition by the term hemizygous. So homozygous, heterozygous, one more is there by the name hemizygous. Hemi means, beta, what happens if suppose an organism is diploid. So normally in a diploid individual, one gene should be represented by two alleles. Suppose if the second allele is missing and only one allele is there, like for example this. So there's X gene which in a diploid organism is represented by only one allele. So in that case, when only one allele is there out of two, then this condition is known as hemizygous. So here the X gene is represented by only one allele, even in a diploid organism. Even in a diploid organism. So this is known as hemizygous condition. What is hemizygous? Hemi means one, right? Homo means both same. Hetero means both but different. I hope all these three terms are clear. Now, apart from this, two more terminologies are there which I am going to use very frequently: genotype and phenotype. Genotype means, beta, the genetic arrangement of a gene in an organism. Like for example, there's a T gene. Now, this T gene can be present in three forms in any diploid organism. Maybe both the alleles are dominant, maybe they are heterozygous, or maybe both the alleles are homozygous but recessive. This is homozygous dominant condition. This is homozygous dominant condition. This is, beta, homozygous recessive condition, and this is heterozygous dominant condition. Correct? Now, see beta, this arrangement of gene, that what is the type of allele of a gene present in an organism, that is known as genotype. So capital T, capital T, capital T, small t, small t, small t is the genotype. Now, because of the genotype, there will be some morphological appearance in the body. That morphological appearance due to genotype is known as phenotype. So what is phenotype? Like this organism will be tall. Here also organism will be tall, but here organism will be dwarf. So tallness or dwarfism is the phenotype. However, their genotypic arrangement, that whether tallness is due to capital T, whether tallness is due to capital T, small t, that arrangement of genes in a diploid organism is known as genotype. So what is phenotype? Tall, dwarf, these are phenotype. What is genotype? This is genotype. Clear? Yes or no? Okay, very good. Now beta, one more thing I want to tell you, which is going to be extremely useful when I will teach you the role of Mendel, that what Mendel worked and how he did. Mendel used one term called pure line. Dekho, beta, there are two terminologies, character and traits. Character means height of a plant. Trait means tallness, dwarfism. So character is basically the nature of an organism, right? Trait means that the two distinguishing characters of an individual is a trait. Like eye color is a character. Suppose there is gene 1 which controls eye color. So gene 1 is controlling character eye color, which can be now either blue, black, brown. So these are the traits. Understood? Now, trait can be of two types. One homozygous dominant trait, one homozygous recessive trait. So these homozygous traits are actually called as pure lines. So what are pure lines? Pure lines are basically homozygous individuals. Pure lines are basically beta homozygous individual. Like for example, height is a character. Height is a character of an individual which can be either tall or can be either dwarf. So these two are the traits. Now, tallness can be due to two reasons, either homozygous tall or heterozygous tall, but pure lines are only when we are talking about homozygosity, not heterozygosity. So tallness due to this and dwarfness due to this condition, this genotype. So what are pure lines? Pure lines are homozygous organism for a specific gene, both dominant and recessive. And for each character, there will be two pure lines. Like height of an individual. One pure line is dominant, another pure line is recessive. Tallness, dwarfism. Understanding everyone? What are pure lines? Pure lines are homozygous individual for a given character. Each character is represented by two traits. Okay, now these were the terminologies which you should know. Now we are going to start with the Mendel's history, who is also known as the father of classical genetics, beta. See, it's not that before Mendel there was no sign of genetics and people were not trying to study about genes. But actually the only successful scientist, even before Mendel, there were many scientists like Knight, Gauss. There were many people who were trying to understand that how genes are transferred, how these genes are responsible for controlling the character of an individual. Many people were running behind it. But the only person who got success was Mendel. That's why we talk about Mendel so much when we are teaching or when we are studying about genetics. Right? So that is why history of Mendel is very important to know. He was born in the year 1822 in a small village of Austria. He actually started the experiment left by two scientists, as I said, Knight and Gauss. These two people, Knight and Gauss, they were also trying to do some hybridization experiment on plants. They were trying to understand that how from this plant the genes are transferred to their offspring, but they could not get the success. So these two people, they left their work in the mid. Then Mendel came and he changed the type of organism. He used pea. इन्होंने मटर पे काम किया. He used pea plant and he started the hybridization experiment. He started cross pollination, self pollination. He started all those things. And he did all these hybridization experiments for 7 consecutive years, starting from 1856 to 1863. For 7 consecutive years, beta, he worked on pea plant. And for these 7 consecutive years, he performed some hybridization experiment to try to understand that how genes are being transmitted from the parents to their offspring. Correct? Yes or no? Okay. Now, what are the things which he did? He actually performed some monohybrid. He tried to study the inheritance of one gene independent of another gene, and then he tried to study the inheritance of 2 genes together. So the 2 types of crosses, the 2 types of hybridization crosses which Mendel prepared were monohybrid and dihybrid. For 7 years, he performed the experiment, and in the year 1864, he published his work in the organization in a publishing house by the name Broun Society for Natural Sciences, and the paper name in which he printed his results were known as Hybridization Experiment in Plants. So, Experiments on Plant Hybridization was the paper name, and which got published in Broun Society for Natural Sciences in the year 1864. So he was born in the year 1822. He worked for 7 years from 1856 to 1863. He actually did some hybridization experiments on pea plant and then finally published his work in the year 1864. And then see how or what were the works which were done by Mendel. Okay, now before we start with the work of the Mendel, we should know what were those genes which were selected by Mendel for his study purpose. Now, Mendel beta chose 7 genes controlling 7 different characters located on 7 pair of homologous chromosomes. So wait, first let me add a page. Okay. Yes, just a second. Right. So basically beta, Mendel chose 7 characters of pea plant controlled by 7 genes. So Mendel chose 7 characters controlled by 7 genes located on 7 pair of homologous chromosome for 7 and he performed actually these experiments for 7 consecutive years. So which is the lucky number for Mendel? Of course, 7. Right? Now, what are those 7 characters chosen by Mendel? Okay, now each character is represented by 2 pure line. Is represented by 2 pure line. Pure line means one homozygous dominant and second is homozygous recessive. Correct? So that means for 7 characters, how many pure lines did he obtain? He obtained 14 pure lines. And to obtain pure line, selfing is required. Continuous self-pollination is required to obtain the pure line. So you can write this in bracket. Continuous self-pollination is required for obtaining pure lines. Right? Chalo. Now let's come back to the 7 characters which were studied by Mendel. Here we are going to write the character and here we are going to mention the pure lines. So here we are going to mention the dominant pure line. And in this segment, we are going to mention the recessive pure line. Now, first I'll be writing all the 7 characters. First character chosen by him was stem height. Then, flower color was the second character. Then, flower position, whether it is axillary or terminal. Then, pod shape. Pod is the fruit. Pod color. Then within pod there are seeds. So seed shape and seed color. So these were the 7. 1, 2, 3, 4, 5, 6, 7. These were the 7 characters chosen by Mendel to start his work of hybridization in the year 1856. The 14 pure lines which he obtained for each character were stem height, tall, recessive, dwarf. Correct? Flower color. Dominant flower color is violet. Recessive is white. Flower position. Dominant is axillary position. Recessive means if the flower terminates, terminal flower. Pod shape. If it is very beautiful, inflated, constricted, पिचका हुआ. Right. Pod color, if it is green, then this is dominant. Recessive, yellow. Seed shape, round. Recessive, wrinkled. Correct? Then seed color, yellow, dominant. Green, recessive. So these were the 7 characters and 14 pure line chosen by Mendel to start his work of hybridization. Hybridization simply means performing cross-pollination, then self-pollination in order to see that what is the pattern of gene transmission from the parents to the offspring. Okay, understood till here? Very good. Now beta, see, I told you there were many scientists who came before Mendel also who tried to identify the nature of gene, to identify the pattern of inheritance, but they could not succeed. So why Mendel got the success? What are the reasons behind the Mendel success? How Mendel could decode the law of inheritance? The first and the very important reason that why Mendel got the success is he chose the right organism to work upon. He chose pea plant. Choosing pea plant, Pisum sativum, was one of the biggest reasons for Mendel's success. Why? Why? First reason is beta, in pea plant, the number of chromosomes are limited to 7 pair. See, lesser the number of chromosomes, lesser will be the complexity. Lesser the gene, lesser the complexity. Okay. Second, per mating, per cross-pollination, per pollination, these pea plant, they produce large number of offspring. So that means sample size will be larger. For one generation, this Mendel will have so many organisms to study. For example, there are 20 offsprings obtained for one generation. Suppose he started working on one, got damaged. Then again he has the second sample now. Started working, got damaged, can move to the third, fourth, fifth. So he had large number of sample size. As a result, this also added to one of his reason for the success. Third, beta pea plant shows self-pollination by showing bud pollination. What? Pea is a bisexual flower. Pea is a bisexual flower. Now what happens? I have already taught you this in our last chapter. One of the contrivance of self-pollination is bud pollination. So when the flower is in its bud stage, at that time only pollination takes place. Hence, when selfing was required to be done, Mendel had to do nothing. It itself was performing self-pollination. Only Mendel had to take care for cross-pollination. When self-pollination was required, Mendel was not worried about it. So his workload got reduced because pea plant itself was showing self-pollination due to bud pollination mechanism. Correct? So these were certain reasons associated with pea plant. Apart from choosing pea plant, the second good reason that why Mendel got success, beta, Mendel's thinking was way ahead of his generation. Mendel could actually realize that he should use the concept of maths and statistics to solve the pattern of inheritance. He used mathematical tools, statistical tools to solve the genetic problems. This concept, this use of maths in biology, see, scientists thought that genetics is a branch of science, and according to Mendel's time, the other scientists used to think that using maths in biology, in science, is absolutely madness. However, Mendel did not believe this. He said, so what? If biology, if it is science, if it is bio, then in bio also mathematical tools and statistical tools can be used. So this helped him to get results faster and better. Apart from this, beta, as I also said, he used to maintain large sample size. So chance of error also got reduced. Plus, he was very vigilant in his work. Vigilance is, of course, very, very important. So these were certain reasons that why Mendel got success and why other scientists of his era who were also working in the same field could not achieve the same success. Clear? Chalo, let's write. Reason for Mendel's success. Number 1. He chose pea as an organism for study purpose. Now, why pea? So, pea is a bisexual plant showing bud pollination. Correct? Second, number of chromosomes in P is limited. Number of paired homologous chromosome are limited, which is actually 2n is equal to 14. Only 7 pair. See, we have 23 pair, 46 chromosome. So many chromosomes, so many genes, so many complexity. How the person will study? He will always get confused in the results. See, pea plant produces large number of offspring per mating. large number of offsprings per mating. Correct? Second reason, Mendel used concept of stats. Mendel used concept of stats and maths in solving the inheritance pattern, the gene pattern of inheritance. Correct? Next point. Mendel always used to maintain. He used to maintain large sample size to avoid or to reduce the chances of error. He used to maintain large sample size which reduces chance of error. Clear? So these are the certain reasons associated with Mendel's success. Now we are going to start with the work of Mendel. But before we start with the work of Mendel, that is his monohybrid cross, first we are going to see the pattern, the how gametes are produced from a diploid individual. So now we are going to study formation of gamete. So before we start with the Mendel's work, we have to see how gametes can be formed. The simple formula to calculate or to form gamete is 2 की power n, where n is the degree of heterozygosity of gene. Degree of heterozygosity. Like for example, beta, there are two genes. One is T gene, one is A gene. In a diploid organism, both the genes are represented by a pair of allele and both the genes are present in their heterozygous state. Can you see? Capital T, small t, capital A, small a. So how many genes are there in this question which are present in heterozygous state? 2 genes. So that means the value of n will become 2. Formula is 2 की power n. So 2 की power 2 means total 4 gametes will be produced. This is how you calculate the number of gamete formation. Second example, this is example number 1, example number 2. Now, here I have given you 3 genes, right, present in a diploid organism. Each gene is represented by a pair of allele. But if you see over here, my dear students, A and B are heterozygous. However, C is present in its homozygous dominant state. So, total number of genes are 3. But for gamete formation, only 2 genes will contribute, that is A and B. C will not contribute in gamete formation according to the formula. So here the value of n will again become 2 because only 2 genes are present in heterozygous state A and B. C is homozygous, you are not going to count. So 2 की power 2, again 4 gametes. Third example, this is the condition. So here in my next example, I have given you only 1 gene and that too present in homozygous state. So value of n over here will become 0. 2 की power 0 means 1 gamete will be produced. How many gametes will be formed? Only 1 gamete from this third example. Right? Now I hope you have understood that how to calculate the number of gametes. Now you have to see that what are the nature of these gametes. Here, what are the 4 gametes? What are the 4 gametes? What is the one type of gamete? So now we are going to see the nature of gamete. Beta, to check the nature of gamete, there is simple branching method, tree branching method. Suppose this is my case. First case, let's calculate the type of gametes. You know, total 4 gametes have to be produced. But what are those 4 gametes? Now I am going to show you. Okay, what happens, beta? This is one gene, this is another gene. So first bifurcate the genes. T gene, one allele, another allele, correct? Now, A gene, you have to write the constituents of A gene under the T gene. See how? Capital A, small a. Again over here, capital A, small a, like this. Understood? Now see what are the gametes. One gamete is capital T, capital A. This is one of the gamete. Second gamete, capital T, small a. Third gamete, capital T, small capital A. And fourth gamete is small t, small a. So these are the 4 types of gametes produced by an individual having these 2 genes, that is heterozygous T and heterozygous A. Have you understood this? Now let's try to check it for the second example. My second example is heterozygous for 2 gene and homozygous for 1 gene. So how will you calculate? You know, total 4 gametes will be produced. See, A gene, bifurcate them. One allele on one side, another allele of the gene on another side. Take the second gene given, bifurcate them as we did previously. One gene, one allele of the gene, another allele of the gene. Here also. Now, third gene is given C, homozygous dominant. Bifurcate it like this. Both are same, dominant. Under every branch, you have to keep on increasing the branches. Right? Understood? Now, beta, see the gametes. Now, this is gamete number 1. That is capital A, capital B, capital C. This is gamete number 1. Now, check the gamete number 2. This is the second branch. This is gamete number 2. Now, you can see, beta, that both gamete 1 and gamete 2 are same. So, you will count as 1. See, both are same, na? G1, G2 are same. So, you will count them as one type of gamete. Okay, next gamete. This is the next branch. Right? Capital A, small b, capital C. This is G3. Fourth branch. Again, in the fourth branch, you will find the same type of gamete. So, G3 and G4 are counted as one type of gamete. So, you have so far obtained 2 types of gamete. Now, let's check the another sets of gamete. This is one branch. Okay. So, small a, capital B, capital C, G5. Another branch. Again, same gamete. So, G5, G6 will be counted as one type of gamete. So, 3 types of gametes you have obtained so far. Now, next branch. This is another branch. A, B, C. And in the last also you will get the same type of gamete. So, here this is the 4th type of gametes. So, 4 types of gamete. How many types of gametes we have to obtain? 4. Now, what are the 4 types of gametes? Capital A, capital C, capital B, then this, and then all small. Correct? Yes or no? So these are the 4 types of gamete obtained in this question. I hope what are the types of gamete, how to obtain them is now clear. You take more examples, try to solve them. One more example I gave you. Let's solve for that. Now that was very, very easy. What is the condition? The third example. Only one gene, that too homozygous, dominant, homozygous condition. So total number of gamete will be 1. And the gamete will be only capital A. This is the only gamete possible from this condition. Clear? Yes or no? Understood? Now let's try to study the monohybrid cross. It is inheritance of one gene at a time. Now beta, what Mendel did? He chose 7 characters. Correct? Now, each character was first studied by him independently. Like first he made a cross for stem height. Like he started cross-pollinating, then he started obtaining offsprings, he started self-pollinating, but studying only one character at a time. So he chose 7 characters. That means how many monohybrid cross will he make? 7 monohybrid cross he will make. At a time, he will study only inheritance of one gene. So basically, monohybrid cross is inheritance of one gene at a time. So monohybrid cross is a cross made with the help of one pair of contrasting trait or with the help of one character. Contrasting trait or with the help of one character. So monohybrid cross is made with the help of one pair of contrasting trait or one character. Now see how he did this. Therefore, Mendel prepared 7 monohybrid cross with the help of 7 contrasting characters which he chose. Monohybrid cross. So Mendel prepared 7 monohybrid cross. Now, beta, whenever cross, maybe monohybrid, dihybrid, trihybrid, whatsoever cross is, the first step in cross, first step of hybridization is cross pollination. So he chose 2 pea plant. One which he considered as male and another which he considered as female. Now beta, see, male flower means, male plant means you are taking the androecium. Female flower means you are taking its gynoecium. So the step number 1 in making monohybrid cross is cross pollination. What is the first step? Cross pollination between the selected parents. Right? Cross pollination between the selected parents. So now he is going to select a male plant and a female plant. For example, we are making this cross for T gene. We are making this cross for T gene. Okay, beta, one more thing. Initially, the parent chosen are the ones who are pure line. That means if T gene, then one of my parent will be homozygous dominant, another will be homozygous recessive. Pure line parent are chosen. So cross pollination between the selected parents which are pure line for a character. Which are pure lines. So suppose I have chosen one male flower whose androecium I am going to use. Female flower whose gynoecium we are going to use. Right? Understood? Yes or no? So, in case of P beta, he chose 2 plants. Male plant, female plant. The plant whose male gametes he is going to consider. The another flower, the plant whose female gamete we are going to consider. Now what is going to happen? First of all, beta, this is diploid. In this diploid cell, you know meiosis will take place to produce the pollen grains, which are haploid, and inside the pollen grain, male gametes will be produced. So, the haploid gametes have to be produced. Now, you know the mechanism of formation of gamete. We are studying it, this monohybrid cross for T gene. So, initially, my parent is capital T. Maybe my male flower is tall. And my female flower, the phenotype is dwarf. That means small t. Right? Now, how the gametes will be produced? Now, both are homozygous recessive. So, number of gametes produced can only be 1. In this case, it is going to be T gamete. Here, the gamete is going to be recessive T allele gamete. Understood? Now, gametes for F1 generation we have obtained. So, now, what are the gametes of first generation? F stands for filial. G stands for generation. So, we have just started our cross. We are going to obtain first set of offspring. So, the generation we started with is the parents. Now, this is the first generation as offspring which we are going to obtain. The gametes from the male is this and from the female is this. Now, what will happen? The gametes will be made to fertilize. Zygote will be produced. Right? Now, these all zygote will be made to develop. Then what he did? He obtained seeds. He sowed the seeds and then he grew all the offspring. What he found, beta? He found, see, this is the genotype which you can see because I am drawing it for you. But actually, Mendel could not see the genotype. He could only observe the phenotype. So, all F1 generation offsprings were tall. So, Mendel, suppose 100 offsprings were obtained. So, all 100 offspring seems to be tall. Now, Mendel was worried because the two characters which he fused with the help of parent were tall and dwarf. See, mother was dwarf, father was tall. Now, father is giving tall character according to Mendel. Mother is giving dwarf character. Now, why not any of the offspring could come dwarf? Why all the offsprings are tall? Mendel was highly confused that how come all the 100 offsprings in F1 generation are tall? Why not even one can come dwarf? So just to understand this, Mendel then tried to perform self-pollination. So after obtaining all 100 offsprings in F1 generation, now what he did to understand that where are the dwarf organism? Where is the character given by mother gone? Only father character is coming. See, father was tall, so all offsprings are tall. Has mother not done anything? Has mother gamete did not give any allele? If given, then where is the character? Where is the phenotype of mother? Why only father phenotype? To understand that, with all 100 offsprings, he performed self-pollination. So now, step number 2 is self-pollination. Now, in this step, Mendel had to do nothing. He had to just wait and watch. Why? Because P is a bisexual flower. It itself will perform self-pollination. So, Mendel did not have any headache of performing self-pollination. Self-pollination. Now, what will happen in self-pollination? Same flower is acting as a male, same flower is acting as the female. Simply cross self-pollination is taking place. Now, what is the genotype of the same parent? So, same parent giving both the gametes, giving both male and female gamete because parent is bisexual. Right? Now, what is the genotype? This genotype. Okay. Now, for this genotype, beta, selfing has to be performed. So this is the anther condition. This is the ovary condition. This is the diploid condition of anther, right? This is the diploid condition of ovule or ovary, whatever you want to write. Now what will happen? Inside anther, meiosis will take place to produce haploid gamete. Now here one gene is there but present in heterozygous state. So the number of gametes produced will be, see, now gametes of F2 generation. Now we are going to obtain the next generation. First generation offspring obtained. Now second generation offspring of F2 generation. So one parent anther का क्या था बेटा? What was the combination? Capital T, small t. So what is the possibility? 2 की power n, 2 gametes possible. What are those 2 gametes? One with capital allele, one with recessive allele. Same goes with ovule also. Inside ovule, meiosis will take place. How many gametes will be produced? 2 gametes will be produced. Now we'll make a Punnett square to obtain the zygotic combinations. So these are the gametes. Now for zygotic combination, for offsprings, to check what will be the nature of arrangement of gene in the offspring, for zygotic combination, we'll make the Punnett square, right? Male, female. These are the gametes from male. These are the gametes from female. Unfortunately, same type of gamete. Now just fill this Punnett square. This is one zygotic combination. This is another zygotic combination. Third, fourth. So how many zygotic combinations we obtained? 4 combinations are obtained. Correct? Now 4 zygotic combinations can you see? But actually out of which 2 is same. 2 zygotic combinations out of 4 is same. Right? So total 4 but type is only 3. Right? One you see as homozygous dominant, one as homozygous recessive, and 2 as heterozygous dominant. Beta, but Mendel could not see all these combinations. He initially did not know the genotype of the organism when he obtained F2 generation. Suppose in F2 generation also, suppose in F2 generation also 100 offsprings are obtained. How many offsprings? 100 offsprings are obtained. Suppose. Now what he saw that out of 100, 75 were tall and 25 were dwarf. So for phenotypic ratio, F2 generation, the phenotypic ratio which Mendel saw was 3:1. He was happy that at least in F2 generation, some plants could gain the female character. See, in F1 generation, where is it? See, in F1 generation, all were tall. They all were tall, 100% tall. No dwarf plant. So here the phenotypic ratio will be for F1 generation. Phenotypic ratio will be 1, where all are tall. So Mendel was highly upset that how come mother is not doing anything? How her phenotype is not even reflecting in one of its offspring. So to ensure this, he performed self-pollination, and after completing it, he observed that 3:1 ratio of the F2 generation, where if suppose 100 offsprings were obtained at last, then 75 were tall and 25 were dwarf. So this only he could see. He could only see the phenotypic ratio. Now he wanted to know the genotypic ratio also. Now beta, you know, for dwarf, there is no issue. Dwarf simply means both the alleles of the gene are small. But in tallness, there are two conditions. Maybe homozygous dominant, maybe heterozygous dominant. So Mendel was now interested to know that if here all organisms are tall, then what is their genotype? Mendel wanted to know if here 75 are tall, then what is the genotype? So this is how first of all Mendel performed monohybrid cross. Till here I am going to take today how Mendel calculated genotypic ratio and what were the laws proposed, what did he understood with the help of this cross. All the things we are going to discuss in our next lecture. But before I end my class, let me just revise the monohybrid cross. Monohybrid means when cross is made for studying one contrasting character or one pair of contrasting trait. So Mendel, because he chose 7 characters, hence he could make 7 monohybrid cross. To start the cross, first step is choosing the parent. One you consider as male, another you consider as female. Then what is done? Cross-pollination is done. Desired pollen grains are made to dust on the desired stigma, right? But before cross-pollination, obviously I have shown you gamete formation. Now one more thing, initially the parents are always taken pure line. If we are considering T gene over here, then male is going to be tall and female is going to be dwarf. Homozygous tall, homozygous dwarf. To produce homozygosity, continuous self-pollination is required. Now what he saw Mendel, he saw that all the offsprings or F1 generation were tall. He did not know the genotype. He did not know that whether the offsprings are homozygous tall or heterozygous tall. He only saw that organisms are tall. He was very upset that why mother character is not reflecting in the offsprings. So he tried to obtain one more generation. But this time he chose self-pollination instead of cross-pollination. So once he did self-pollination, he obtained the phenotypic ratio of the next generation as 3:1, where 75% of the offsprings were tall and 25% were dwarf. So this time Mendel was happy because he could see female character also in the offspring, maybe in the second generation. Now, the genotype of dwarfism is clear that okay, small t, small t. However, now Mendel wanted to decode the genotype of F1 offsprings and 75% of F2 offsprings which are actually tall. He wanted to know that whether they are homozygous dominant tall or they are heterozygous dominant tall. Right? So other phenotypic ratio for monohybrid cross in F1 and F2 clear to each and everyone. Now we are going to talk about the genotypic ratio but in the next class. So with this I would like to end my today's class while giving you some homework. You have to solve these questions as well as many more questions which you get from outside. Try to solve them till here and we are going to proceed further in our next class. So thank you for watching me. Keep studying, keep working hard. Yes, all the best. Bye-bye everyone. Take care.